Hello everybody, my name is Jacob Avery, and I'm going to talk to you guys a little bit about the Song Lake zooplankton during fall turnover. So, a little bit about me and the project. I'm currently a student at SUNY ESF, and I am taking a limnology practicum course. And this is just a practicum course about inland waters, specifically lakes. And for a field trip for this class, we visited Kofokla, or the Cortland Onondaga Federation of Kettle Lake Associations. And one of the leaders of one of the lakes expressed to our group that she's very interested in learning about the zooplankton population in that lake, which happens to be Song Lake. And so me and my partner were interested in this, so we decided to do a study about the zooplankton assemblage during fall turnover versus during summer stratification. So a little bit about Song Lake. It is located about 25 miles south of the city of Syracuse in New York, and it is very close to the town of Tully. And you can see the red star on the map there is where Song Lake is located. So a little bit about the watershed. As you can see, this red outline here is the Tully Lake watershed, which encompasses all of the kettle lakes that are in this kettle lake group. And then you have in the blue outline the Crooked Lake watershed, and within that Crooked Lake watershed you have the Song Lake watershed, which is very small. It's actually only around 1.5 square miles. So I've been calling Song Lake a kettle lake, but what does this actually mean? It's just a term to describe the way it is formed. So about 10,000 years ago, when the glaciers came through, as they pushed forward, a piece of it got broken off and buried by the glacier into the soil. And then when they receded, that buried chunk was left there, and then it eventually melted and left a depression in the earth, which filled with water, and that is how a kettle lake is formed. And Song Lake has no inlet or outlet because of how it was formed. So, I also mentioned I mentioned stratification. I'm going to give you a little bit of background on what thermal stratification is. So, if you don't know, during the summer, the lake actually separates into layers. And most lakes, it's 3 separate layers. You have the epilimnion, the metalimnion, and the hypolimnion. And this happens because the water on the top interacts with warm air during the summer. And because of this, it heats up the top layer of the water, which becomes less dense than the rest of the water, causing that water to stay on the top. And then all the water on the bottom is much colder and more dense. And during the summer, when the lake is stratified into layers, these layers actually do not mix at all. So you have 3 separate layers that don't interact with each other whatsoever, which is kind of cool. I never knew that. But when the air temperatures start to decrease during the fall, overturn happens, and this happens because the cold air is interacting with the epilimnion, the top layer of the water, and this cools that top layer, which increases the density of that layer. And once it hits a level where it's able to mix with the bottom layer, when they're at the same density, these layers will mix, and the whole entire lake will mix, and the layers will be broken down. So, why does this matter? What are the effects of thermal stratification? Well, if there's no mixing during the summer, there is, towards the latter half of the summer, there will be less oxygen, or sometimes even no oxygen, in the hypolimnion, which is the bottom layer. And this happens because during spring mixing, the hypolimnion gets cut off from the oxygen supply at the surface. And as organisms are respirating down there and using up oxygen, there's a certain oxygen budget. So as the time goes on, organisms use up that oxygen and it kind of depletes. So we get a whole different kind of ecosystem down in the hypolimnion compared to water at the surface of the lake. There will also be a lower temperature in the hypolimnion. That is also because it is not interacting with the warm air that is above the lake. So, what are zooplankton? Well, let's break down the word. So the prefix zoo just means animal or a living being. And then the suffix plankton just means organisms that drift, float, or move passively in the water. So these are basically just small organisms that can't really swim against the current, and they just are kind of at the water's mercy on where they go and where they end up. So these guys are usually less than 2 millimeters long and often microscopic. You can't see them with the naked eye, but some of them you can. So a little bit of diversity in zooplankton. There are 3 main groups that pertain to freshwater. You have your copepods, and this group isn't too diverse. Most of them look like that, but there is a few variations. And as far as size ranges go, they can range from very small to very large, sometimes the largest that you'll find in the lake. And then below that you have the rotifers, which is a very diverse group. Some of them look like the one in the picture, some of them look completely different, some are just blobs. It's a very diverse group. These guys are often the smallest out of all of them. And then lastly you have the cladocerans, or some people call them water fleas. And these guys are somewhat diverse, and for the most part, most of them are pretty large. And just look at that guy, isn't he cute? No, they're not cute. They kind of look like little aliens that are swimming around in the water. So, what are the importance of these zooplankton? Well, most of it boils down to what they do in the food web. They're kind of a link to the primary producers to the top of the food chain. So they're kind of right in the middle. They're important in keeping the phytoplankton or algae populations down, as well as being a really good food source for small bait fish like shad. And they're also a main food source of juvenile game fish. So if you were to take zooplankton out of a lake, you would not have any fish because they're a really good food source for fish, and you would also see a very large algal bloom because nothing is eating the algae. And they also act as an indicator species. You can tell a lot about the lake by what's in there and how big the zooplankton that are in there are. So now that I give you a little bit of a limnology lesson, let's talk about the project. So me and my partner looked at the zooplankton distribution during stratification versus fall overturn. So on this map of Song Lake, during summer stratification, we went to the blue point, which is the deepest spot in the lake, and we took samples from the epilimnion, the metalimnion, and the hypolimnion. And then during fall overturn, we went out when the lake was mixing, and we went to the blue point and did the same exact thing as we did when it was stratified, as well as we went to 4 other points just to collect some other data of the lake as a whole. So how we did this, for the zooplankton collection, we used a device called the Schindler trap. As you can see here, as you're lowering it into the water, the top lid and the bottom lid stay open. And then when you're at the depth you want, you kind of pull it up, and then the lids shut close, and the water that was at that depth is captured in there, and then you bring it up, and it is filtered through that trunk there. So all the zooplankton will get captured in that mesh trunk while all the water is drained. So then you have a filtered sample of all the zooplankton that were in that trap. And then to capture some of the water properties at different depths, we used what is called a YSI probe. And this measures dissolved oxygen, temperature, and depth, and many other things. So yeah. And then once we collected our data, we took it back to the lab. We first added ethanol to kill all the zooplankton because they're much easier to analyze when they are dead and not moving. And then we filtered the samples. So we just put it through this sieve tube here, and just basically concentrated the zooplankton into a lesser volume of water. And then once we filtered them, we took them to a dissecting scope and put them in a zooplankton counting tray so they could be easily counted. And then we identified and analyzed all the data. So just so you have an idea, this is kind of what we're looking at under the microscope. The image on the left here, you can see that really big zooplankton, that there's a copepod. And then some of the other ones, there's some other copepods in there. And then the one a little bit above it is a cladoceran. And then on the right here, we have 2 cladocerans here. And so yeah, that's pretty much what we're looking at for many hours at a time. And some of the acknowledgments that I'd like to thank for this project is, first of all, SUNY ESF, and then also the Cortland Onondaga Federation of Kettle Lake Associations for letting us use your lake as our study site. And I'd like to thank Dr. Kim Schultz, our professor, for helping us with this project, as well as our TAs Aaron Ebert and Jackson Booth. And I'd like to thank my partner, Brandon Chartrand, as well as Ann Moore, our senior advisor, for all of her help along the way with this project. Thank you very much.